Thanks for the opportunity to be here today. My name is Karen Whitkin, and I'm a program director in the Division of Cancer Biology. And I'm here to talk to you about a concept that was developed by the Cancer Moonshot Implementation Team E, which is co-chaired by Malcolm Smith and myself. Oops. Can I have some help from the AV support? Sorry. So the best way to change the slides will be here?
Yes.
Great, thank you. Several types of childhood cancers are associated with chromosomal rearrangements that lead to the creation of fusion oncoproteins, like the EWS-FLI1 that I'm showing you here as an example. In this case, the strong, constitutively active transactivation domain of the EWS-R1 protein is placed in front of the DNA binding domain of the normally developmentally regulated FLI1 gene. The resulting fusion oncoprotein still does what FLI1 does. It still activates developmental programs, but now it does so at an inappropriate time. And this, in a nutshell, is really what happens in a lot of these fusion-driven childhood cancers. You end up with a fusion oncoprotein which does aberrant transcriptional regulation, changing developmental programs, which leads to transformation of a unique cell of origin. The fact that these fusion oncoproteins are absolutely specific for tumor cells, and the fact that they're found in cancers that have few other genomic lesions, suggests that, at least in theory, these fusion oncoproteins could be exceptional drug targets. In practice, however, there have been a lot of challenges to therapeutic targeting of these fusion oncoproteins. The first is that it's just difficult to target transcriptional regulators with small molecules. But above and beyond that, there have been other difficulties. In many of these cancers, there's uncertainty about the cell of origin. There's not a lot of understanding of these fusion proteins, the proteins that they interact with, or their key dependencies. And really, there's just a lack of good model systems for doing the preclinical studies that are necessary to really understand these fusion proteins and develop therapeutics. One approach that has worked, that has held some promise in recent years, takes advantage of the fact that these fusion oncoproteins normally exist in complexes. The idea is that instead of just looking at the fusion oncoprotein itself as a drug target, to actually look to see what is it the oncoprotein is binding to, what's downstream of that, how is it changing developmental pathways, how is it initiating tumors and leading to tumor progression. So to look not just at the fusion oncoprotein, but all along that progression to see if there are potential drug targets anywhere in there. And in fact, this is an approach that has worked quite well in MLL fusion leukemias. The MLL fusion oncoproteins also make poor drug targets. But work in recent years has revealed that they need to bind to the menin protein in order to turn on the developmental program that leads to leukemia. This knowledge has led to the development of small molecule inhibitors that actually target the menin-MLL interface, turn off this gene expression program, and can even cause leukemia cells to differentiate or even enter apoptosis. The Pediatric Cancer Working Group of the Blue Ribbon Panel recognized both the high potential and the challenges associated with therapeutic targeting of fusion oncoproteins, and they identified this as a key area for research acceleration. They felt that this was an area where targeted investment could have an enormous impact. And that's partially or mostly because these fusion oncoproteins are well-credentialed oncogenic drivers of high-risk pediatric cancers, many of which do not currently have promising targeted therapies in the pipeline. This includes the sarcomas, the NUP98 fusion leukemias, the Relay fusion ependymomas. These are cancers that do not have promising targeted therapeutics right now. Above and beyond that, as our BSA subcommittee reviewers pointed out, the fact that these fusion oncoproteins in childhood cancers occur in genomes that are relatively otherwise silent make them sort of a simplified system that provides a unique opportunity for understanding how fusion oncoproteins can cause tumorgenesis in childhood cancers, but also in adult cancers as well. The recommendation of the Blue Ribbon Panel Pediatric Cancer Working Group was to take a multidisciplinary, collaborative, and comprehensive approach to studying these fusion oncoproteins, going from structure-function data to target identification, small molecule inhibition, and all the way to therapeutic testing, and that this should be done in a collaborative approach that took advantage of all of the areas of expertise listed on this slide. The Pediatric Cancer Working Group of the Blue Ribbon Panel went on to identify 5 specific areas of research focus. One was the development of model systems for preclinical studies. 2 was defining the critical dependencies that these fusion oncoproteins create. 3rd was defining how these fusion oncoproteins influence gene expression and change developmental programs. 4th was identifying the protein complexes bound to these fusion oncoproteins and using a structural biology approach to define the domains and the structure of the fusion oncoproteins themselves and also complex members that they associate with. And finally, identifying small molecules that can either inhibit these fusion oncoproteins, their activities, block the critical interactions like we've seen with the MLL menin example, or selectively lead to fusion oncoprotein degradation. Our BSA subcommittee reviewers asked us to put this concept in the context of existing NCI funding opportunities. There are a few existing opportunities from NCI right now that are relevant. One of these is the NCI Pediatric Provocative Questions Initiative. There are also R01 and R21 program announcements out right now for gene fusions and pediatric sarcomas. Both of these predate the Cancer Moonshot initiative, and both provide opportunities for investigators to submit applications for discrete projects that may be relevant to our goals here. The third on this list, the PA 17138, is actually an administrative supplement. This is for current NCI grantees, people who have a current NCI eligible grant, who can now apply for additional funding to form a new collaboration for a project within the scope of their existing grant focused on fusion oncoproteins in childhood cancers. This actually is a moonshot initiative. We put out this PA in FY17 in order to start to build the collaborations that we felt were really needed in order to form the network recommended by the Blue Ribbon Panel. So we feel that these existing PAs, or these existing announcements, are relevant and they're complementary, and now we're ready to take the next step, which is to really go in and put this consortium together that the Blue Ribbon Panel recommended. So what I'm here to propose today is a consortium that we call the Fusion Oncoproteins in Childhood Cancers Consortium. This would be a U54 multi-component program to support multidisciplinary and multi-institutional collaborative teams that take a comprehensive approach to understanding both the basic biology of these fusion oncoproteins and developmental therapeutics. Each team would focus on just a single fusion oncoprotein, and the whole program would be limited to a few high-risk fusion-driven cancers, meaning cancers that don't already have promising targeted therapies in the pipeline. In order to make sure that the teams stay on track, we will be using milestones to evaluate their progress throughout the duration of funding. There will also be an annual meeting as well as other opportunities for engagement with other relevant NCI efforts, and this includes the grantees of a lot of the grants that I just talked about of these other NCI efforts. Here's a little bit more detail on the budget and logistics. This would be an RFA for U54s. There would be a set-aside of $7 million per year for 5 years. There would be single receipt dates in FY18 and FY19. Review would be in a special emphasis panel. Right now, we want to fund 3 centers. The budget would be about $2 to $2.5 million a year total cost each. And there would be a separate set-aside for collaborative projects. This could either be collaborations between these investigators in the network, or investigators outside the network could use these funds to collaborate with this network. There would also be access to shared NCI pediatric core services that we're putting together right now. And this includes things like large-scale protein purification, antibody production, and even access to some structural biology facilities. I wanted to end by mentioning some markers of success for this initiative. We'll know that this initiative has been successful when we start to see some of the recommendations of the Pediatric Cancer Working Group of the Blue Ribbon Panel come to fruition. Things like seeing the development of faithful models of these fusion-driven cancers, seeing the identification of critical dependencies that these fusion oncoproteins create, better understanding of the binding partners and the pathways by which gene expression and developmental changes occur, and finally, the identification of small molecules with therapeutic potential. Thank you, and thank you to our BSA subcommittee reviewers for a thoughtful discussion, and I'm happy to take any questions.
Dr. Shannon.
Yeah, thanks very much. That was really a terrific presentation and summary of both the RFA concept and also the discussion that we had. I think some of the highlights, just that these fusion oncoproteins that dominantly deregulate transcription are clear initiating and driver mutations. They're present at very high allele frequency, both at diagnosis and relapse of these patients, and there are very few other associated mutations. So it's essentially, if we're going to be able to figure out how to drug transcription factor fusions, it seems like a pretty good place to start. The absence of preclinical models, I think, has really held back the solid tumor area as opposed to the leukemia area, where there's been a lot of progress with MLL and some of the other fusions. And clearly, we thought that this, both on the working group and I think the Blue Ribbon Panel endorsed this, the idea that this really would be very well suited to a sort of larger scale multidisciplinary approach. One of the things we particularly encouraged the NCI in writing the RFA to consider is ways of incentivizing and encouraging these groups to come together from multiple different centers rather than from single institutions, because quite honestly, the scientific expertise in these fields is fairly thin and it's spread out. And so you'd really, if you're going to bring chemical biology, structural biology, biochemistry, animal modeling together to attack some of these, you really want the best people in the field trying to do this no matter where they're located. So we were very supportive of this, and we're very pleased to see in the presentation that the NCI staff has made some revisions consistent with the comments of the committee. Chris, I think, was concerned in his comments about how this was distinct from and would interact with other NCI initiatives in this area, particularly the sarcoma RFA that's out for R01s. But I think this is pretty clearly different from what any individual R01 investigator could accomplish. I'll let Chief speak for himself, but there was some question both in the implementation team and the committee about, is this such a narrow focus on these transcription factor fusions, or should we be considering things like overexpression of MYC and neuroblastoma? And I think the decision was to sort of stick with the suggestion of the working group and the Blue Ribbon Panel to really focus narrowly on these very disease and tissue specific lineage transcription factors. But I'll turn it over to Chi.
No, I mean, I'm not going to repeat much of it, but I think this is one of the areas that NCI should definitely push very hard. It's very similar to the RAS program in a sense that you can't kick the can down the road anymore. You got to tackle it and see what happens. So I'm very supportive of it as well.
And I probably should have mentioned that, you know, we haven't really moved the bar very far in terms of improving the treatment for these patients since the 1980s. And particularly when they get combination chemotherapy, radiation, the late effects of these therapies in this young population are the fearsome late effects. So we really need to do better in terms of more molecularly driven therapies. So with that, I guess I can move to get us to our coffee break by voting on this measure.
All right. Discussion. Yeah, no, I'm open up for discussion now.
heard about others trying to make immune therapy approaches to these fusion proteins, because these are neoantigens, right? The definition of a neoantigen is this. So I've heard of other people talking about that before, but I didn't see that as...
Yeah, so Liz and I just discussed that.
Yeah. So it's in the Blue Ribbon panel recommendations under immunology. So we wanted the immunotherapy part to be both pediatric and adult, and we specifically suggested that, but good point.
Yeah, and I'm going to do double duty tomorrow. I chaired, there's a BSA RFA tomorrow. We're going to review about a pediatric immune therapies network. Turns out these tumors probably aren't particularly immunogenic because of their very low mutation burden. So that's a bit of a problem.
Max?
In a way, expanding on that, I think that this is a wonderful opportunity. I was just so struck of kind of how prescriptive this was, in that you defined exactly kind of what they should look at each step and what to target. And although a scientific mind would look at that and say it's very rational ideas, it almost doesn't allow, in my mind, enough freedom for someone to think of something completely different, of a way to target these that may be outside that scope of what you put there. Yeah. And for instance, in developing also animal models, which I think is critical in this, I think one of the areas is cell of origin that's really important, because just knowing these fusions isn't enough. You really have to know what cells to express it in and things. So in other words, there's a lot that I think you should allow more freedom of what comes in and not try to be so prescriptive.
Yeah, I think, we did talk a fair amount about the tissue and lineage specificity. I'm sorry if I didn't emphasize that. I guess my view of this is that the opportunity for the creative young woman or guy investigator who wants to try some pie-in-the-sky idea about some component of this, that's what the RFA for the R01 is. What this is a concerted effort to bring people to the table who think about how transcription factors work, how they transform cells, how they form complexes, how you pull the protein components out of those complexes, how you then develop small molecules. It's a really more of an industrial soup to nuts sort of effort. It may fail, but that's the idea, and the committee was pretty much behind that.
Scott?
As someone who works on one of these fusion proteins that's a transcription factor, I don't really see how this fits into the moonshot, because this is not going to be something that's going to offer a cure in 5 years. This is a long-term basic science problem that you've just laid out. You need to identify proteins that interact, targets, transcriptional networks. I mean, it seems like that's completely outside the purview of what the moonshot was supposed to be, which was to move the translational needle in 5 to 7 years.
So this came out of Peter's subcommittee. Well, I'm not going to answer for you, though. So, I think the group had deemed that this is an unmet need, and until you put major effort in it, something that's going to take 50 years, going to be shortened. Maybe we'll make some advance in 5 years, but no, the deliverables is whether you can achieve the milestones that's been articulated.
I guess, not to draw this out, but I guess the argument might be that since these are tumors that, as you know, are so much of the phenotype is driven by a single oncogenic mutation, and there's likely a very strong cell type dependency, that if you happen to hit a home run and generate a small molecule like a menin or a .1L for MLL, you would imagine you would not be playing the whack-a-mole and pancreas cancer game that you're going to inevitably play even with a good RAS inhibitor, where it's going to come back and come back and come back until you figure it out. You could really have a major clinical impact if you got the right target and the right small molecule in these diseases. So, who knows, 5 years from now what we're going to be thinking about the moonshot, but I suspect you got to try to take a lot of different kinds of shot on goal, and some of them are going to hit and some aren't.
Okay, you heard the discussion, so I'm going to ask all those in favor, unless there are no questions. Any against? Abstention? All right, we get an earned few minutes extra for the break.